Today we're speaking with Dr. Gordon Mills. He is chair of the Department of Systems Biology and head of the Kleberg Center for Molecular Markers at the University of Texas MD Anderson Cancer Center. Thank you very much for joining us, Dr. Mills. Could you begin by talking about the promise of personalized medicine?
So in the past, we have treated all patients pretty well the same way. If a patient comes in with ovarian cancer, Mrs. Green, Mrs. Brown, and Mrs. White all got exactly the same therapy. We know that only 2 out of 3 patients benefit from our best therapy today, and that means that one of those 3, we didn't know which, got a toxic therapy that didn't help her. Personalized medicine is that we will know in advance by studying what's going on in their own genes, their genetic makeup, and what's happening in their personal tumor to know that Mrs. Brown and Mrs. White benefited and should get that therapy, but Mrs. Green needs something else. We also are moving from using the chemotherapy approaches that were designed to act average across everyone to now targeted therapies where we're going after the genetic changes that are specific to Mrs. Green's, Mrs. Brown's, and Mrs. White's tumor. The goal is to specifically target what's going on to get a more effective outcome and hopefully much less toxicity.
And what are the technological needs to realize those opportunities?
So the key now is to be able to characterize every patient's tumor and their own genetic makeup so that we can understand what is the best approach for that individual. Those technological needs include being able to look at all of the things that are happening at the DNA level, the RNA level, and the protein level, and most importantly, how we integrate that information into making a decision that will help patients. There has been an incredible revolution in what we can do to measure changes in DNA and RNA. Protein lags behind. At the DNA level, we can now sequence an individual's complete genome in 2 to 3 weeks. We can sequence all of the genes, or all of the parts of their genes that form proteins, the important parts, in a few days. This is a technological revolution that is incredible. The challenge right now is understanding what that information means. We generate terabytes of data on every individual and have to say what is important and what's not important. What changes drive the behavior of the tumor and would make that patient respond to particular therapies or not others, and which of those are just passengers, things that came along for the ride and we can ignore. So the technology has really improved at the DNA level. Our ability to interpret and understand it still lags behind. At the RNA level, we have been able to measure changes in amounts of RNA for many different genes. Patients are already being treated based on those changes. We also have some very nice studies from that showing who does and does not need chemotherapy with early breast cancer. The Oncotype DX test is one of those types of things that spares women from toxic therapies. We have much greater problems at the protein level. At the DNA and the RNA level, we have a very limited amount of material, a limited number of genes to deal with, and for DNA, everything is there at exactly the same amount. The estimate today is that the average cell has between 1 and 6 million different proteins that can be present at enormous differences in amount, 10 to the 9th fold difference in amount. So finding technologies that can deal with that complexity and the differences in concentration has been a major challenge. There's some big advances that are happening. They're just not coming along anywhere near as fast as what we have to deal with at the DNA and RNA level. The first of those is an improvement in the use of mass spectroscopy. Mass spectroscopy allows us to study proteins. It's the only technology that we have at the protein level that is pure discovery. We can find things that we didn't know about. It's limited, though, to the top 2,000 to 4,000 most common proteins in a cell, and that's nowhere near the 1 to 6 million we need to study. Now, you can make that a little better by purifying some of those proteins up first and working that through, and that is helping, but it's still primarily a discovery technique. Some new technology in mass spectroscopy, something called stable isotope analysis, now allows us to compare 2 samples very well and tell exactly what are the differences quantitatively. And in something that's really new, something called multiple reaction monitoring, for the first time we can quantify a protein where we don't have an antibody for it. It's still relatively low throughput, a little bit expensive, a little bit slow, but it is a tool that we haven't had before. And so these are really big changes that are moving things forward. This is also being moved forward by an effort at the National Cancer Institute called CPTAC. It's the Clinical Proteomic Technology and Cancer Program. And the goal there is to standardize approaches so that we can share information across labs. That's been a problem in the past, but the quality control, standard operating procedures are now in place to move this forward much better than ever before. The other technology that has made a big difference is protein arrays. We're now able to take hundreds, even thousands of antibodies and put them on a single slide and analyze a patient's sample against 1,000 at a time. And that gives us a lot of information, very useful information on one individual. Our laboratory has focused on a different approach, and that's taking thousands of patient samples and putting them on a single slide and then characterizing them across 200 or 300 different antibodies. That has allowed us to discover protein changes that matter in patients' tumors. Going back to the example that I gave you about RNA, we can now identify with very strong confidence, needing additional clinical trials, women with early breast cancer who don't need chemotherapy, who will have such a low chance of recurrence with just hormonal manipulation, which is not as toxic, that they simply don't need and probably won't benefit. We're also using that technology because we can look at thousands of patients across fairly large numbers of important molecules to look for markers that would say patients will benefit from particular targeted therapies or that those therapies are working or not. So basically, what we need is improved technologies to look at what's happening in the patient and the patient's tumor, but more importantly, we need the ability to integrate that information with all of the clinical information and all the other things that a doctor uses to make decisions with their patients and come up with a personalized program for each patient we see.
I want to talk a little bit about Stand Up to Cancer. You're a co-leader for the project targeting PI3K in women's cancers, and the project's goal is to discover approaches that will predict which patients will respond positively to PI3K inhibitors that will accelerate personalized cancer treatment. Can you give us an update on how the project is going?
So the project has just started. We were identified as one of the funded DREAM teams recently, and we have now had 4 different face-to-face meetings as a group. We have at least 1 teleconference a week and innumerable emails back and forth. What has happened is that a group of individuals, really incredible individuals, not just in our DREAM team, but in all of the DREAM teams, that had not collaborated together before in a major way, have been brought in to form teams. These teams really are incredible. The goal was to put together a set of people who could dream, who could make a difference, and bring together the best of the best across institutions to collaborate. And that's happening. Not only have the DREAM teams come together, but we've got all of the teams in a group to build a DREAM team of DREAM teams. And that communication back and forth is really, I think, starting to make a difference. The other part that's come out is that even in the short time that our DREAM team has been in existence, we've done a lot. We have already characterized one of the 3 women's tumors in more patients than have ever been characterized before in more depth around this PI3 kinase pathway. In one of the 3, endometrial cancer, we found a brand new aberration, a change in the tumors that will alter the way the patients respond to therapy, and that happened since we started the DREAM teams. So they really have brought together a group of people who can and will make a difference.
And lastly, could you remark on the innovative grants screening process and the caliber of investigators that Stand Up to Cancer funds?
So the innovative grant program was to look for the next generation of scientists. Most of the DREAM team leaders have mentors in their program or mentees in their program where the senior people will help bring some young people along. We need that next generation of outstanding people to benefit and to utilize all this new information. We reviewed a very large number of applications, one-page applications to say who would be likely to be exciting. 80 of those were asked to submit applications, and they were incredible. But was even neater is 18 people were invited to Philadelphia to present to the committee. And I can tell you that of those 18, I would have hired almost every one of them on the spot if I had an opportunity. This next generation of bright young kids that are going to grow up and fulfill this promise of personalized medicine, taking ahead all of the new genomics, were really incredible. I think what was even more exciting is that many of these people were physician scientists, people who were both clinically qualified, making a difference directly by working with patients, and also doing the research that's going to help the next generation. The other that was really striking is how much support they had from their institutions. These were not people who had just been hired and put in a corner and said, figure out how to do this. They had support, they had people who were helping them learn how to do research, and more than that, the institutions had set up an environment where they had access to all the tools, all the technologies I've been talking about, so that they could figure out how to move forward. It was truly an enjoyable and exciting process. The 13 that were funded were just absolutely stellar.
Great. Dr. Mills, is there anything that I haven't asked you that you'd like to touch upon?
I think that one of the things to bring forward with the idea of personalized medicine, the dream teams, and where we're going, is that we already do a degree of personalized medical care. We already treat breast cancer patients based on the changes that are occurring in their tumors, and we know that that treatment, targeting patients with the right type of therapy, hormonal therapy if they have estrogen receptor positive therapy, HER2 therapy or HER2 targeted therapy if they have an amplification, chemotherapy for patients with other types of breast cancer, is making a difference. We see this in lung cancer with the EGFR targeted therapies. We're seeing it in some rare diseases like GIST with targeting KIT, and even some new exciting stuff in melanoma targeting particular genetic aberrations. It's not a matter of saying, are we going to be able to do personalized medicine in the future? It is, how personalized are our approaches going to be? Can we really go from treating the average patient and getting an average outcome to doing something that's going to be very specific for each individual? We're talking about the future. It's always risky when you try and predict the future, but we are doing personalized medicine, and it's going to just get more and more so in the future, and continue the fact that fewer and fewer people are dying of cancer every year. I think this will be a big step in keeping that happening.
Dr. Mills, thank you very much.
Okay.